everyone. I'm Terre Constantine. I'm the Executive Director and CEO of Brain Research Foundation, or BRF for short. I would like to welcome everyone to this special presentation on a very timely topic, anxiety and anxiety-related disorders. I'm thrilled to introduce our speaker for this evening, Dr. Denise Icahn. And before she starts talking, I just wanted to tell you a little bit about her relationship with BRF. 2 years ago, Dr. Icahn received a Brain Research Foundation seed grant, which is a startup grant BRF awards for innovative research projects. This was the first grant she received as a junior faculty at Mount Sinai. She just recently finished the grant in I believe June 2020. And we're pleased to report that the BRF funded project led to approximately $6 million in additional external funding for her to continue her research. That's a really impressive return on BRF's investment. And I want to thank you for doing that, Denise. She'll be talking more about her research and how it relates to anxiety. Her presentation will be about 20 minutes, and then we will have 5 to 10 minutes of Q&A following her talk. We're so pleased to have her with us tonight, and I'm going to turn it over to Dr. Icahn. Thank you.
Hi, Terry. Thank you so much for having me, and thank you all for attending. And I'm really excited to share some of the work that we're doing and also discuss with you and answer your questions. I just want to first off, let me actually share my slide here. One moment. Okay, so I want to first just say thank you so much to Brain Research Foundation. As Terry said, this was the very first grant I got as a junior faculty member, and I think it really made a big impact in my research career in at least 2 ways. The first was, I had applied to several grants already prior to applying to the Brain Research Foundation, and I got a lot of no's. And some of the feedback I got was, this is an interesting idea, but there's no reason to think that the brain behaves this way. And that we just didn't have enough preliminary data and it was too risky of a project. And so getting the research funds from BRF to do this risky project means that we were able to not take the safe route, but that we would try something new, and that if it worked, it would have a really good impact in understanding how trauma memories are processed in the brain. But I think the second way that it really influenced my career is maybe even more important. So I think it gave me the confidence I needed as a young investigator to pursue science at the highest level. So when I started my professorship here, I'm a mother of 2 young children, and I had a 3-year-old and a 5-year-old, and anyone who has young kids know that that takes a lot of time, and there's a lot of sacrifice both ways to do work and taking care of the kids. And when I looked around, there weren't a lot of other women scientists who had, really made it in their careers, and there are even fewer women that had children. And so I really was questioning whether I could do this job or not. And after so many rejections, I thought, maybe this isn't the right job for me. But having that first grant come in from BRF really gave me the pat on the back and the confidence I needed to pursue the high impact science that I set off to do. So thank you so much to BRF, and I'm very excited to present some of the findings that we've made with this funding source. So first, I just want to acknowledge all of the people and our funding agencies. So as Terry mentioned, that BRF was the very first agency to fund us. And since then, many other people and many other foundations have followed suit. And so now we're super excited that we can continue the work, the questions that we started asking with BRF, funded through other foundations. So thank you to our funding agencies, and thank you to my fantastic lab. I have to say, this is one of my favorite slides, and because the reason I love my job, the reason I have the best job in the world, is because every day I get to come and work with generous, brilliant, creative, kind people who are really passionate about understanding the brain so that we can translate our results and better develop treatments for neurological disorders. So my lab is really interested in how is it that we store memories across our lifetime. And one of the amazing things about our brain is the ability to accumulate what seems like infinite number of memories, and somehow we can file and sort them in all the right places in the brain such that at a later time we can recall that memory accurately and use it in the future. And so here's just an example of some pictures of memories that my 6-year-old daughter Clarity has collected in her, already 6 years of life. And one of the really important questions that drives my lab is how is it that we process trauma memories? How are these stored in the brain? And here is just an example of my daughter a month ago during the global pandemic. She had a freak accident, fell off the ladder and ripped off a part of her finger, right? Pretty traumatic experience. So how is it that she and we store these types of trauma memories in the brain? So there is a lot of people who study trauma memories, and one of the things that motivates us is because the majority of the population will experience a traumatic event at one point in their lives. And a lot of time when we talk about trauma memories, of course, we think about the war veteran, right? So military trauma is a big type of trauma, but there are many types of trauma: sexual assault, child abuse, vehicular accidents, natural disasters like the wildfires in California and on the West Coast. And let's not forget COVID-19, right? Many people, because of the global pandemic of COVID-19, are experiencing trauma, and the list goes on. And we know that experiencing these trauma have really important implications for psychiatric functions. For example, trauma can, some people who have trauma will later experience PTSD, a debilitating anxiety-related disorder in which the trauma memory will be recurring such that it makes it very difficult for functioning in the everyday life. We also know that trauma can potentiate anxiety, depression, and even trigger substance dependence. So understanding how is it that our brain is able to store trauma memories so that we can then maybe potentially manipulate these trauma memories and mitigate some of these consequences would be really important and impactful. So how are memories, including trauma memories, stored in the brain? So the perspective that I come from is looking at kind of at the circuits level. So there's a lot of research that suggests these trauma memories are stored in a subset of cells in the brain, and we call these neuronal ensembles. And that when we recall this memory later on, such as here, my daughter trying to remember being at the hospital waiting for her surgery, she might reactivate many of the same cells that were previously activated when she encoded that memory. And so this idea that many of the same cells are being reactivated suggests to us that these memories are very stable and stored in a stable representation across these many cells in the brain. And if she were to recall a different event, such as here, climbing on her grandparents' tree in their yard, we would expect that a different neuronal ensemble or different cells in the brain would be reactivated because different memories are stored in different cells across the brain. So how do we study this, right? So I don't actually look into my daughter's brain and measure her cell activity. My lab uses an animal model. We study this in mice. And while I was a postdoc at UCLA, I got to work with a really fantastic team in which we developed an open source miniature microscope. So we took, one of those big tabletop microscopes that you might see in your biology class, and we threw away all the non-essential components and miniaturized it so it's about the size of my thumb. And we can take this tiny little camera, which is made of camera sensors that are in your cell phone, and we can put it onto the mouse's head, and the mouse will wear it like a hat. And while the mouse is running around and exploring and interacting with the world, creating memories, we can peer into the awake and active brain and look at which cells are active and representing the memories that the animal is forming. So then I just want to spend 1 minute to talk a little bit about this technology and the importance of the BRF funding. So one of the challenges in getting funding is getting funding to support not only the innovation of the technology itself, but the dissemination of the technology. And given the flexibility and the support of the BRF, we were able to use some of the funds and go to different parts across the world and take this technology, and we held workshops in New Zealand, in China, and Israel. This is before COVID-19, of course. And they would say to us, you know, without us going to teach them how to build this technology, they would have to pay something like $250,000 for this technology, where we taught them how to make it for $1,000. And so this was really breaking a barrier of access to research. And we wanted to, you know, not other labs be limited by their funding, but that they would have accessibility such that they can really answer the questions that are going to make advances in understanding the brain. So thank you to BRF for that. But back to the science. So going back to the questions of how memories are encoded. So while we don't look in humans in my lab, we are looking at in mice. So let me give you an example of the type of experiments that we do. How do we see the memories in the brain? So we put on these little cameras on the mouse's head, and we'll bring the mouse into a new environment. And here we give them a shock, okay? And it's a foot shock. It doesn't cause any permanent damage to skin or anything like that. But this now pairs a very aversive experience with being in this novel environment. And we can see that this memory is encoded into a sparse neural population in the brain. And many people, including work from us, have done these types of studies. And then when we bring the animal back, we don't present the shock. But without presenting the shock, we can see the animal recalling the memory, because many of the same cells that were activated during the memory encoding are reactivated while the animal's recalling. And we can know by the animal's behavior that because through their defensive behavior showing a fear response. So in this case, if animals are afraid of this environment because they remember being shocked, they freeze. And we can see that the level of freezing, a proxy for memory, seems to scale with the level of reactivation of the prior neural ensemble. And then when we put an animal in a different context, one that they've seen before, we see that different cells are representing this memory. And so this is how we study it in our animal model. So you might ask, if it's really the subset of cells that encode the specific trauma memory, if we silence those cells, can we erase the trauma memory? And so we and others have done this, such that there's a trick that we use called optogenetics. It's just a fancy word for being able to manipulate the cell activity. And in these types of studies, we and others have shown that if you silence the cells that represented this context, then the animal no longer remembers this memory and no longer expresses fear of this context. And we think that this has really important implications for treating anxiety-related disorders or PTSD, right? If we can somehow separate the trauma from the memory itself, then maybe we can mitigate some of the aversive symptoms of PTSD. So these are ideas that many people currently are exploring with patients. So on the flip side, if these cells are really underlying the memory of being shocked in this box, then if we bring the animal to a different box, ones that they've never been shocked, if we then somehow, again, using this trick of optogenetics, we shine light into the brain and we can selectively activate only those cells that represented the shocked context or this blue context that was aversive, we can see that the animals will trigger this fearful memory and also exhibit freezing response or proxy to being shocked in a context, despite that they've never been shocked in the context that they're in. So together, these studies suggest that memories are encoded in these neuronal ensembles and that we can bidirectionally manipulate the memories. Now, this is really exciting. However, in a lot of these animal studies, we are studying how animals remember single memories. But in the example that I showed you, right, real world memories don't happen in isolation. As we experience the world, we're constantly learning about different things and we're constantly experiencing and forming memories, such as in this case. And the question we wanted to ask with the BRF funding was, how are trauma memories linked with other memories? Now, why is this question we were even interested in any way. So let me give you a kind of intuition of why we thought this was important. So let's recall, here my daughter has, a very traumatic experience. Her finger came off, and here she's in the hospital waiting for her reconstruction surgery. And presumably that memory gets encoded in a subset of cells we call the neural ensemble. Now, it would really behoove my daughter if she thought back and said, why am I here? Why am I missing this finger? And she thought back to when she did not listen to her parents and she was climbing that homemade ladder that she was not supposed to climb. And if somehow she was able to link those two memories together, right? If she thought of one, she would think of the other. This would allow her to make better decisions, in the future. Oh, 1 second. Okay. So we wanted to see if the brain was able to do this, right? Not only code memories in isolation, but link experiences with the past so that we can make predictions about the future. So the way we tested this in animals was we put an animal into this neutral context that was safe, and we looked at the neural activity and saw that there was a neural ensemble that was specific to this red box. And then a couple days later, or hours later, we brought the animal back, or not back, but we brought the animal to a new context in which we gave them a strong foot shock. And now we paired this traumatic experiences in this box. And we saw that this memory was encoded in a different neuronal ensemble. Now what we did was we followed the animal while it was sleeping in its home cage. And what many people's work would suggest is that they are not only sleeping, right? Their brain's not totally shut off, but they are reactivating many of the cells that were active during the trauma experience. And so this phenomena that we call neural replay, the replay of the neural ensemble during sleep, we think is a mechanism that is really important for stabilizing these cells to represent the memory. So that was not surprising at all. That's what many people have found in the past. What was really surprising was that not only was the memory of what just happened reactivated, but in addition, it was as if the brain was searching for something in the past that can be related to this traumatic event to be linked together. And that the reactivation of not only the trauma memory, but a prior novel experience, such as here this red context, got linked together during this offline period while they were sleeping. And we can test this to see if the memories were actually linked, because when we brought them back into the red context, remember, they've never been shocked in this context, but what we saw was that when they recall this red context, it triggered the recall of the shocked context. And so even though they've never been shocked in this context, they exhibited fear, suggesting that these memories were somehow linked together. Now, if it's really the reactivation of this neural ensemble underlying this, memory of the red box, then if we silenced the reactivation, right, silence these cells during sleep, we asked, could we then separate the two memories such that when they came back to the red context, they no longer exhibited fear, because thinking about the red context no longer triggered the memory of being shocked in that blue context, which is exactly what we saw. So these preliminary data suggest to us that sleep may be really important for this process. Now, what I just presented to you, right, red context, blue context, it might be a little confusing. And so let me just give you a summary of kind of the intuition for how this might occur with our memories. And so the question I asked was, how are trauma memories linked with other memories, right? How do we link what just happened to the past so we can make predictions about the future? So based on this preliminary data that we collected, supported with BRF funds, what we think is going on is that after trauma experience, this memory gets encoded into a subset of neurons in the brain, and that during sleep, not only is what just happened reactivated, but it's as if the brain seems to be searching for related events to link together. And potentially this may be a circuit mechanism in which allows us to link the past to predict the future. Now, we think this has really important implications for anxiety-related disorders like PTSD. So in this case, what I described to you is quite adaptive. You want to link things that are causally related. But you can imagine a case, the trauma is so pervasive and so strong that not only are you linking what just happened with maybe one event that's specific, but now instead of my daughter linking it to the ladder that was causal, she might be linking it to climbing on the tree or rock climbing or all different types of climbing. And this would be very maladaptive, that seeing anything that relates to climbing would trigger a fearful response. And so these are questions that we're continuing to explore with other grants that we received since receiving the BRF. So the take-home message, 3 simple points. Memories are stored in neural ensembles across the brain, and these memories can be selectively manipulated such that we can suppress the memory or activate the memory. And from our preliminary data, it suggests that aversive experiences are linked with past memories during sleep, and this can be adaptive or potentially also maladaptive. So once again, I just want to thank my lab for the incredible work that they've done, and my very first grant from BRF that really, set the trajectory of my lab, both in the science that we do and having the confidence in the way we do our science. And thank you to all the other funding agencies for supporting this work and continuing this and related work. So thank you so much, and I'm very happy to take questions.
So thank you so much for that interesting talk. And I'm happy, very happy, and BRF is too, that you decided to stay in science and that we were a part of it. So congratulations. I think these are really interesting findings. And what we're going to be doing is opening it up for Q&A. So if you have a question, those that are watching, please type it in at the bottom of your screen where it says Q&A. You can even do it anonymously if you don't want to submit your name. In the meantime, I'm going to ask a question while we see some things hopefully coming in. So you said about, well, first let's just say, how is this, what can we do during COVID right now in this pandemic to hopefully not overlink or over, stress when you're right. They tell you to sleep, and now I'm thinking maybe I shouldn't be sleeping so much. I don't know. So,
yeah.
What would you do for, or what would you tell the people that are
Absolutely. So sleep is, it's a really interesting thing. So actually sleep deprivation has been tried on human patients after undergoing trauma like war trauma, and that has gotten mixed results. But in general, I would say that, we also have data showing that if you don't sleep, right, so you don't actually remember the bad things well, such that you might actually then generalize to not the memory of the bad thing, but other things as well, because you don't have a specific memory of the bad thing. The other thing about sleep is that, sleep is a big predictor of depression, anxiety, and so not sleeping actually triggers anxiety and depression, and particularly in people who are susceptible. So I would say absolutely sleep and try to get the best sleep hygiene so that, for mental health and our memory systems. And the other thing related to COVID, sleep has been shown to, well, if you don't sleep and if you're sleep deprived, you will actually have a deficit in your immune system. And right now, we want to have the strongest immune system we can to fight against COVID, the flu, that season's coming up. And so I would say sleep is a good thing.
All right, well, that certainly makes sense. Let's see, Jill is asking, has the mini scope been adapted for humans? And is the manipulation of the neuronal ensembles been successful in people? Has it been successful in people? Yeah, I wrote that down too. You said they were doing it in people, but I was like, how are they doing that?
Right. So let me step back and kind of describe a little bit about this. So the mini scope itself has not been used in humans, although I am collaborating with Mark Baxter, a colleague at Mount Sinai, to try to get this working in monkeys, and other people are also trying to get it working in non-human primates so we can get one step closer. So one thing that people have done in humans, and I've done this in part collaboration with Sarah Bennick, but looking at using fMRI, looking at kind of more global imaging. So in that way of imaging the brain, we can't see single cell activity like we can in our mouse studies, for example, but we can kind of see global changes across the brain. So multiple clumps of cells and how they fire. And using machine learning, people can do similar types of analysis to see that similar clusters of activity in the brain are specifically representing a phobia or a trauma memory compared to other types of memories. And so that's one aspect of looking at the neural substrate of memories or trauma memories. Now, in terms of how do we apply the type of technique of like memory erasure to humans, right? Memory manipulation. So one of the ideas is that if you recall the memory, that memory now goes from something that's stable to something that is labile and can be manipulated. And so one idea is that if we recall the memory and we take a beta blocker, for example, or something that reduces stress, now that memory of that trauma will be associated with reduced stress or reduced anxiety or reduced visceral responses. And so that is something that's being tried clinically, and the parameters for how to get that work for different patient populations is very, very specific. And so there's been mixed results, but a colleague here, Daniela Schiller, is working actively on that to see exactly under what conditions for human patients who have PTSD would that kind of manipulation work.
Okay, really interesting. Dan is saying, great job presenting a complex story very quickly and in a way that everyone can understand. Can you specify which brain region or circuit that you're imaging from?
Yeah, so we were specifically looking at the hippocampus, which is the brain region that is really important for storing kind of what we call these episodic memories or experiential memories. But these kinds of findings have also been observed in other brain areas like the amygdala, which is really important for encoding fearful experiences or highly emotionally salient experiences. And so now we're actually taking a brain-wide approach. And so with the mini scopes, we can only look at one brain region at a time. And so now we're using a new technology where that instead of taking kind of like a video camera of the brain active, we can kind of take snapshots of a couple of different time points of cells across the entire brain. And this technique is called iDisco. Funny thing is actually my daughter named Clarity was named after this brain clarification technique that allows you to clarify the brain and see all the cells that are active. So she's not only in my talk, but also in my scientific influence.
Oh, pretty interesting. Dave is asking, and this is a really good question, what do we know about anxiety unrelated to trauma? Just plain old being worried or uncomfortable about one's environment. I don't see COVID worries the same as like losing a finger. So what's the difference?
Yeah. So, I think that stress and anxiety, so anxiety, I would say, is more is on one end, but a lot of studies have shown that some level of stress is important, right? And that if you're totally comatose, you're not going to learn anything, you're not going to be able to function. And a little bit of stress is actually good for encoding memories, for performance, attention, all of these really important processes. But too much stress can really impair your ability to perform, to remember, to hold anything in your working memory. So there's what we call inverted view, that little stress is good, optimal, and then too much stress can be really bad or even debilitating. And so, but constant stress. So what I talked about is kind of an acute stressor, right? One-time event. And that's kind of what we study. But a lot of other people study chronic stress. And how does chronic anxiety change maybe your ability to remember, to react? And so I think little stress is good, too much stress is bad.
And kind of on that same vein, it seems like some people are more resilient than others. Is that true? You know, so I look at my kids and they're doing okay during COVID. You know, they figured it out, but I know others are really having a hard time with it. And so, you know, what's the difference? And is it easier for kids versus adults, maybe?
Yeah, that's a really
or the other way around?
Yeah, that's a really great question, and a very active, you know, line of research right now. And so we know that there are biological differences, whether it's in your DNA or if your parents, if your mother had stress during gestation, that can also influence a child's predisposition to stress. So there are a lot of factors that influence your predisposition to stress. So if you have previous experience with substance abuse or had a prior trauma, that can also make you more susceptible to enhanced levels of stress later on. But you know, what's really interesting is that across a lot of research, including my work, we have found that some of the biggest predictors, the most reliable predictors of stress and anxiety is social isolation, right? That's like how we induce stress in our animals, and unpredictability. And I would say right now, we've really been living under a lot of social isolation and unpredictability. And then if I can just add one more thing, is sleep disruption. And so which, you know, because a lot of us are not going outdoors, we're not getting natural sunlight, and also there's just stress, right? Not allowing us to sleep normally. So I would say, you know, in terms of practical tips, right, to avoid social isolation, to reach out to your community, connect with somebody. And I know that it's different from men and women to connect, and I definitely see it in children, that my girl loves to Zoom and talk with all her friends, but my boy was really sad during this time and did not feel like he can just Zoom with his friends, because that's not what 8-year-old boys do. And so for him, it was playing video games, and then all of a sudden they just couldn't stop talking, playing video games, strategizing, right? So so I think also now, as we're thinking about children, they are also being social isolated, right, with not being able to go to school. And so finding ways for them to connect in a new way, I think, is going to be one of our challenges.
No, I think that's all right. And very similar to my children too. I have a boy and a girl. I'm going to ask one more question, and our audience is very lovely and they really want to know, how did your daughter's surgery go?
Oh my gosh, it went so well. I am so grateful for plastic surgeons. Who knew that they can reconstruct a finger from a different body part? It's amazing. And my daughter is doing fantastic, and every day she tells me how great life is, and she likes to tell me all the reasons why her life is so great. And so I think, you know, if anything I can learn from her is how she's so resilient, and that can influence how I can stay resilient as well.
Well, that's great. I want to put up one more slide, if I can find it. We are going to have our virtual discovery event on October 29th, 2020. I hope everybody can attend. There will be a panel discussion on autism for that evening. And for more information, you can go to the website that you'll see up there. And also, I believe that they may be chatting out a link to go to the event page. So I hope that everyone will, I hope everyone enjoyed the talk, learned something. I certainly did. And I hope that you will also turn in for the benefit on October 29th. And I want to say again, thank you so much, Denise, for joining us this evening. And I'm so happy that you were one of our grantees and that you've done so well.
Thank you so much, Terry. And thank you so much, everyone, for your attention and your questions and your support of BRF and brain research.